Hi, good morning. So today we're going to move deeper into our lecture on the TCA cycle. And actually today we're going to finish up. What we covered last time was talking about linking glycolysis with the TCA cycle. And we're starting out with describing that glycolysis. It's awful. Glycolysis is in the cytoplasm. And at the end of the 10 steps of glycolysis, we have our pyruvic acid, pyruvate, which I'll give the structure of here. And like I say, this happens in the cytoplasm, but the next steps of aerobic respiration, they occur in the mitochondria. And because they occur in the mitochondria, that's across a cellular membrane. And so you need a transporter to pull across that cellular membrane. And the steps that are going to happen in the mitochondria are going to be the TCA cycle and oxidative phosphorylation as well that comes out of that, which we're going to utilize the NADH and NAFADH2 that are generated in glycolysis and in the TCA cycle. We're going to use those in oxphos. But first of all, we're going to feed that pyruvate into the TCA cycle because there's still some energy in this molecule. There's still some energy in some of these bonds, especially here with this methyl group. And we're going to, so the TCA cycle is trying to try and get some of this energy out of it. So first of all, it needs to be, the pyruvate needs to be transported into the mitochondria. And there's a mitochondrial pyruvate carrier that's going to carry it across that membrane. That's those two different compartments. And then there's going to be the reaction by the enzyme PDH, pyruvate dehydrogenase. And that's going to result in the loss of CO2. So if we have our pyruvic acid molecule, you can see that there's a really obvious CO2 that can come off. And you get loss of CO2, which comes off. And at the same time, what you generate is the acetyl-CoA. And in the end, you end up, let me highlight it. This is an acetyl group. So in the end, you end up with acetyl coenzyme A. The loss of CO2, the reaction of the thiol of coenzyme A with the carbonyl carbon of this acetyl group, and a reaction catalyzed by the enzyme PDH, generate acetyl-CoA. And you also generate through that this, there's an oxidation which is occurring. This molecule is becoming oxidized. And I've been saying that these oxidation reactions get coupled with reductions. And so the reduction is from NAD plus to NADH. So we're going to be generating some NADH here, which is going to be useful for our to feed into oxphos, as I've been describing. Now, if I can generate a new page. Oh. I'll just move this all to the side. Now, where we move on to next are the 8 steps of the TCA cycle. We start out with oxaloacetate, which has got 4 carbons, and we take that acetyl-CoA and we feed it in, and it's 1, 2 carbons, because remember this has come off. So you have this 2 carbon acetyl-CoA, and they condense together to form the 6-carbon citrate. Citrate or citric acid. That's going to go around, continue to go around. You're going to get one CO2 come off, where this citrate oxidizes and get a loss of CO2. You get a second CO2 coming off, where the molecule gets oxidized. And the first time it gets oxidized, the CO2 comes off, and it now becomes a 5-carbon chain. Second CO2 comes off, and it becomes a 4-carbon chain. And it's going to continue cycling around the 8 steps until you get back to oxaloacetate at the beginning, which can then react with another acetyl-CoA in a condensation reaction to generate another citrate and go around again. And the point of this is that in doing so, you generate 3 NADH at 3 different steps, which is going to be useful for feeding into oxphos. You also generate an FADH2, which is going to feed into oxphos. And you also generate as well GTP, which can be swapped for ATP. But that's something that we'll move on to when we go through these steps in detail later on. But the whole point is that this cycle can go around indefinitely, because one oxaloacetate can, in theory, cause the oxidation of an infinite number of acetyl-CoA molecules, because this oxaloacetate condenses with acetyl-CoA to form a citrate, goes around the cycle being oxidized once, releasing CO2, twice releasing CO2, generating that NADH. There's other steps as well, but I'm just kind of condensing. And then again restoring oxaloacetate to do the reaction again. So it's just going to keep going around and around. But the whole point is that you generate this NADH, this FADH2 in particular, and also this GTP, because the NADH and the FADH2 is going to be utilized in oxidative phosphorylation to generate a very large amount of ATP, significantly more than in anaerobic respiration, fermentation of glycolysis. So this is that's the rationale behind it all. And I'm now going to go through all of the steps step by step. A small thing right at the end of this lecture, which I'll mention now and I'll mention at the end as well, is that there are reactions which feed into and pull out of the TCA cycle. And the TCA cycle is a major hub of metabolism where multiple things can feed in and multiple things can feed out. So, for instance, there may be certain metabolites such as alpha-ketoglutarate, which I'll abbreviate as alpha-KG. And alpha-ketoglutarate is one of the metabolites in the middle of the TCA cycle. It's a 5-carbon metabolite, and it can you can exchange, oxaloacetate, alpha-ketoglutarate is just the keto acid of the amino acid glutamic acid, glutamate. And if you take the amine group of glutamate, you generate the keto acid alpha-ketoglutarate, or if you add the amine group to alpha-ketoglutarate, you generate glutamate. And there's an enzyme that can do this. There's several enzymes that can do this. You can go backwards and forwards to add an amine group or remove an amine group. And so you can add to the TCA cycle, and adding is anaplerism. Oh, I ran out of space. Or anaplerotic reaction. And to take out is cataplerotic, as in ana, to build, kata, to break apart. So anaplerism, you're adding to the TCA cycle. Cataplerism, you're taking out of the TCA cycle. And there's a few of these. You know, a really classic example is alpha-ketoglutarate can shuttle in and out. Sometimes you need to make amino acids. sometimes you break down amino acids to supply energy. So you can go back and forth between them. There's a few of these. One of them in particular is actually to go from when we have pyruvic acid, and we contribute pyruvic acid in the anaplerotic reaction through acetyl-CoA, which I mentioned. You know, the pyruvate dehydrogenase causes the oxidative decarboxylation of pyruvate to turn it from this 3-carbon metabolite to this 2-carbon metabolite that is reacted with acetyl-CoA, reacted with cofactor A to generate acetyl-CoA, and it feeds in as a 2-carbon. But also, this 3-carbon pyruvate, you can add a carbon to it to generate the 4-carbon oxaloacetate. So you can actually add a CO2. You can remove a CO2 and add it as acetyl-CoA, or you can remove a CO2 and add it as acetyl-CoA, or you can add a CO2 and add it as oxaloacetate. And that is again a anaplerotic reaction. Now, the reasons for that are something that we're going to get into in the more detail with the next series of lectures when we talk about gluconeogenesis. And this is a reaction that actually requires ATP. So it's an energy-consuming reaction to generate oxaloacetate. But sometimes it is necessary. Sometimes it's necessary to restore metabolites to the TCA cycle. But more importantly, it's actually important to go from pyruvate back to glucose. And you can't go, I'll call it glucose, so you don't mix it up with glutamate. You can't go directly from pyruvate to glutamate, just straight up the glycolysis. Instead, you need to convert that pyruvate into oxaloacetate and then go up through glycolysis. And that may seem complicated and confusing. We're going to get into it in the next series of lectures, but I did want to just mention it. And really, to summarize, we start out from the last lecture. We've generated pyruvate from glycolysis. That pyruvate is transported into the TCA cycle by the mitochondrial pyruvate. My mind's gone blank. Mitochondrial pyruvate carrier. There we go. I'm sorry. And it's transported, and then within the mitochondria, pyruvate dehydrogenase, the complex we talked about last time, catalyzes a reaction, the oxidative decarboxylation of pyruvate from a 3-carbon to a 2-carbon acetyl-CoA. In doing so, you, of course, generate CO2. And that's some of the CO2 that we exhale. That cofactor A reacts with the acetyl group to generate acetyl-CoA. And you also, that's an oxidation that is coupled to the reduction of NAD to NADH that can enter into the oxidative phosphorylation. And that 2-carbon acetyl-CoA can enter in the TCA cycle where it condenses with, and I specifically use the word condense, condenses with oxaloacetate, which is a 4-carbon metabolite. The 2-carbon and the 4-carbon condense together to create synthesize the 6-carbon citrate molecule. This is going to go around the other steps of the TCA cycle. And there's 2 oxidative decarboxylations where, again, you lose CO2, and so it becomes 1 carbon shorter. But each time you reduce NAD to NADH. There's another NAD reducing step as well. There's an FAD reducing step to FADH2, which can also enter into the TCA cycle, very similar to NADH. And we're going to talk about that in a few weeks as well. You also do generate a GTP, which is easily converted into an ATP. There's 8 steps in total. When you add to the TCA cycle for whatever reason, that's anaplerism. When you take out, that's cataplerism. One of the main reactions of that is of the transamination of alpha-ketoglutarate to generate glutamate, and you can go glutamate to alpha-ketoglutarate or alpha-ketoglutarate to glutamate. You can go back and forth. And another key anaplerotic reaction is adding a CO2 to the 3-carbon pyruvate to generate the 4-carbon oxaloacetate metabolite as an anaplerotic reaction. And one of the main reasons for that is in gluconeogenesis. But gluconeogenesis is something that we're going to go on to a little bit later, but I just wanted to set the scene a little bit now. So we're going to go through the 8 steps of the TCA cycle. There's a couple of them which I go into in detail, but many of them I just go into in short amount because I just want to focus on some key things. We can't focus on all of the reactions in too much detail because there's just so much to cover. We could do a whole 16-week on just on the TCA cycle, but we're just condensing it down to some key points I think are particularly important. So I'll start the lecture material now. Oh, here it is. Okay. So I'm not sure which screen you guys are going to be seeing. I'm going to swap screens, and so you can hopefully this is the correct scene here. So like I said, we start out with the 4-carbon oxaloacetate and the 2-carbon acetyl-CoA, and they condense to form the 6-carbon citrate. There's 2 oxidative decarboxylations, which generate a 5-carbon and then a 4-carbon metabolite. But each time, because that oxidation is coupled with the reduction of NAD to NADH, which are going to feed into oxphos, which generates ATP. There's also a step where you generate GTP, which is can be interconverted to ATP, and an FADH2, which behaves very, very similarly to NADH in the context that we're using here. It's different, but in this way it behaves the same. And a third NADH as well. But as you go through these steps of this 4-carbon, 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 eventually you end up with back at where you started, your 4-carbon oxaloacetate, which can then react with another 2-carbon acetyl-CoA to regenerate your 6-carbon citrate. And you can go around again and again, an infinite number of times, as long as you don't lose your oxaloacetate and you keep supplying with acetyl-CoA. So let's move through to the slides. So this really is just describing that all of these reactions take part in the mitochondria. And the citrate, which is formed from the condensation of acetyl-CoA and oxaloacetate, you generate the CO2 from the oxidative decarboxylation and these useful energy metabolites. Many of this, there's going to be two different kind of, we can break down these 8 steps into 2 groups really. An energy conserving oxidation, so that's the one which is going to generate one of these metabolites here, but also moving a isomerization, second type isomerization, where, well, not necessarily isomerization, but kind of in some way rearranging the molecule. I mean, there's like a hydration as well, which definitely is an isomerization, but something which is going to be easily facilitated, a rearrangement of some kind, which is really putting things into position for, number one, the energy conserving oxidation to occur. And a lot of this is really generating carbonyl groups, because carbonyl groups are more reactive than methylene groups or methane groups, or alkenes or alkanes. And so you generate carbonyls instead often with this step 2, which makes it easier for the step 1, a energy conserving or type 1 energy conserving reaction to occur. It's important to note that a lot of these metabolites, a lot of these enzymes rather, in the TCA cycle are proteins and protein complexes which are grouped together, such as a complex 8, 1, and 2. I mean, there's probably more as well, which are close together. They're going to be all close together in a large supramolecular complex, And this really facilitates the easy transport of metabolites between the proteins, the enzymes, rather than just taking up a substrate and casting out the products into the unknown. It can just shuttle it to its friend next door. Makes it much more efficient and easier to use. And these kind of grouped together complexes, these grouped together enzymes, we call them metabolons, to make these really huge multi-enzyme complexes. So I kind of gone through this already. The oxaloacetate and the acetyl-CoA generating citrate, going around, generating NAD, and there being two oxidative decarboxylations. I'm gonna go through these steps in detail now. As a lot of people like to group step 2, which is catalyzed by the enzyme aconitase. And a lot of people like to break that down to a step 2 and a step 2A and a step 2B. I generally don't. I just consider it as one reaction together. So don't get mixed up there. Let's just, for the purposes of my lecture, let's just call this step 2. Let's not do 2A and 2B. Okay, so let's move through the steps. So first of all, we have the formation of citrate, and that is, like I said, the condensation of acetyl-CoA and oxaloacetate to generate citrate. There's this 2-carbon and 4-carbon to generate 1, 2, 3, 4, 5, 6-carbon citrate. You can see that right away the biochemical delta G is quite large and negative, which is telling us that this reaction really favors going forward, going to the right a lot. Part of that is because in the reaction, the oxaloacetate concentration is pretty low. So there's there's not very much on this side, and there is a lot on this side. And so generally, in many equilibriums, that might favor moving in this direction. But the equilibrium in this case favors going to the right, despite the imbalance in metabolites. And so this is really important. I'm going to go through the mechanism of what happens here with the intermediate citrulls CoA, coenzyme A, and I'd like you to, I'm going to condense the steps really and summarize the steps, but I would like you guys to know them, and these steps are something that will be tested on in an exam. So first, it's important to know, I've talked about before the induced fit model, and we have the induced fit versus the lock and key. And really that the lock and key is something that you may be taught early on in biochemistry and biology classes, but the idea that it's actually a lot more complicated than that, and you need to factor in the induced fit model. And the induced fit model is the idea that substrates binding, as they bind, both the protein and the substrate will change shape. And in doing so, they make it easier for each other to bind to each other, but sometimes for other things to bind. And so there's a case of the induced fit with citrate synthase, enzyme number 1. Enzyme number 1 of the TCA cycle, citrate synthase. In that you have these two, substrate 1 plus substrate 2, reacts together. Let's go back. Substrate 1 plus substrate 2 plus the enzyme, so can't forget the enzyme, come together to form the enzyme substrate 1, substrate 2 complex, which are then going to react to give you the enzyme product complex that is going to break apart to give you enzyme plus product. So that's really the reaction. But they need to bind in a specific order because there's two things that can bind, and it's actually important that they bind in the right order. Otherwise, they won't be orientated properly within the protein, within the active site of the enzyme. And that's actually, that's on the next slide. But it needs to be in the order of, if this red curve here is the active site, a little pocket within the protein, it needs to be substrate 1 and then substrate 2. It wouldn't work if it was substrate 2 and then substrate 1, in that order, it wouldn't work because you can't. They're not orientated correctly for the reaction to occur. It needs to be substrate 1 and then substrate 2. And in order to facilitate that happening, oxaloacetate binds first, and then what needs to happen is that oxaloacetate needs to bind first, and then acetyl-CoA, so that they are properly orientated within the enzyme. And in order to make sure that happens, the shape of the enzyme is not super complementary to acetyl-CoA until oxaloacetate binds. So if acetyl-CoA comes along to the enzyme which hasn't bound to anything yet, the acetyl-CoA will come along and it won't be able to bind because it won't be a good fit. But the act of oxaloacetate binding to citrate synthase causes a conformational change in the protein, and that makes it so that it then will then be complementary to bind with acetyl-CoA, so it can now come in. So it kind of forces the order to be correct, because substrate 2 won't bind until substrate 1 binds, because the binding of substrate 1 changes the protein to make it bind substrate 2 better. Now, the mechanism is given by these on these next 3 slides. And I'm going to summarize it really quite simply, and you will be tested on the summary of how what occurs. And really, I can break it down into 3 steps. The first step, and we can see here that this blue shading is the enzyme, and this His 320, His 274, ASP 375, aspartic acid, histidine. These are referring to the 3 letters, of course, refer to the amino acid, and the number refers to the number of amino acid within the primary sequence. And so these are catalytic residues involved in the catalysis of the reaction. And you can see that oxaloacetate, of course, is bound first. And this is an acid catalyzed aldol condensation. And you should remember aldol condensation from OChem. And really, it's the in the acid catalyzed aldol condensation. Essentially, what happens is that you can see actually here. This is where it's happening. Is that the an aldol? Oh, sorry, I'm sorry to keep jumping back and forth. This is an aldol. This is where you have the this carbon has an alcohol group attached to it, a hydroxyl group, so alcohol, but also a alkene. So this is an aldol, and this aldol, you can see here, can come in and attack a carbonyl carbon. Because this carbonyl carbon is slightly delta positive and it's acting as a nucleophile and can come in and attack there. And so in step 1, I kind of group several things. You can see that you can see with all of these arrows, there are lots of things happening at once. I'm just going to condense it down quite simply, and there's a text description here that's kind of also summarizing, but also a little bit more full than what I'm going to say with my words. But in general, what is happening is that this carbonyl is getting protonated. So there's your acid catalyzed. This is getting protonated, and doing so that causes a rearrangement to generate the enol. And the electrons in this double bond are going to act as a nucleophile to attack this carbonyl carbon here. So that's really step 1, is the acid catalyzed generation of the enol intermediate, which is going to attack the enol intermediate of acetyl-CoA. And that is going to come in and attack the carbonyl carbon of the carbon number 2, 1, 2. Of the oxaloacetate. That's step 1. Nucleophilic attack. And that's, I mean, different textbooks and different professors will explain that in a different way. I'm just summarizing it into 3 basic steps. There's a lot going on here. The second, so this is that this here is this here. So we're just continuing from where we were before. That's the same, showing the same step. You now have generated this. There's now a rearrangement, and you generate this citral COA. Well, this is kind of like citric acid here, where this is really essentially citric acid. It's kind of like citric acid COA. Now, it's not citric acid COA. It's structurally very similar to it. That's what I'm trying to communicate. They're structurally similar, but it's citroyl COA. And so the difference here is that this isn't a carboxylic acid down here. Instead, it's this thioester with the cofactor A. So the second is the first step is the nucleophilic attack, which generates the citroyl COA. And then you get this, and then, sorry guys, I've messed this up a little bit. The nucleophilic, so it's the first one is the sorry, I've messed up the way which I'm describing this. First of all, it's a protonation, which is the protonation, the acid catalyzed protonation that I described. And then it's the nucleophilic attack. I condensed these all into step one. I apologize. I got muddled with the video lectures throw me off. So it's the protonation, which allows for the nucleophilic attack, and then you get the hydrolysis of the cofactor A that releases the cofactor A and generates the citrate. So step 3 is the hydrolysis of the CoA, cofactor A. So you no longer have this thioester. Instead, you have a carboxylic acid, which is citrate. So you go from the citroyl CoA to the citrate. To summarize, step 1, you get the protonation. Step 2, you get nucleophilic attack of the carbonyl of oxaloacetate. Which is the condensation. And then step 3, you get the hydrolysis of citroyl CoA to citrate citric acid. So I'd like you guys to be able to recall these 3 steps, to recall and briefly describe these 3 steps. That's that's important. Cool. Now we'll move on to step 2. So we've generated this citrate. We've got the oxaloacetate and the acetyl CoA to citrate. Next step is going to be a rearrangement of citrate to isocitrate. Step 2. Step 2 is the rearrangement of citrate to isocitrate with this enzyme aconitase. It is, I mean, as with everything, it's a reversible reaction. See, the biochemical delta G actually slightly favors the reactants. But again, with these things, it's going to be pulled forward by the low concentration of product, because the product is going to be shuttled into reaction number 3. So if there wasn't a reaction number 3, then the equilibrium would balance with a lot more, I've drawn that the wrong way, with a lot more reactant than there is product. But because the product is constantly being moved, the equilibrium is going to seek to replace it by LCP, Lachetti's principle. And so even though the reaction doesn't really favor the, the biochemical delta G, that's where if you started out with an equal weight on the balance in the equilibrium of 1 molar product, reactant and product, it would actually slightly favor reactant. In the biological setting, because of the low concentration of product, the reaction gets pulled to the right towards product because the product is being used for step 3. There's not really much I want to say about this. We're just rearranging from the citrate to the isocitrate. Let me just clear up the screen a little bit. Rearranging from the citrate to the isocitrate. There's this aconitate intermediate in the middle. I generally don't talk about it too much. You get the dehydration and then the hydration. So the dehydration generates the alkene and then the hydration of the alkene. So you've just moved this hydroxyl group from 1, 2, 3 to 1, 2, 3, 4. You just move the hydroxyl group from carbon 3 to carbon 4. And you dehydrate, generate the alkene, rehydrate, generating the alcohol again, but it's the different position. And that's really all there is to it. And I don't want to go into too much detail about that. I'd rather focus on other enzymes. But just to briefly mention is that part of the complex of this happening is that in order to stabilize this reaction, there is this iron sulfur cluster, which is being held in place by cysteines of aconitase in a complex. And these iron sulfur clusters are going to come in important later. They're just reactive centers. And you can see that they're forming a variety of different bonds, coordinate bonds and hydrogen bonds, the holding everything in place. I think something that I just skipped over briefly with enzyme number 1 is that notice in several cases we have these hydrogen bonds which are helping to hold things in place actually. And it's the same thing with this enzyme where the iron sulfur cluster is helping to hold and orientate the substrate in place. Many of the amino acids, the residues, and the cofactors help to orientate the substrate into the active site of the enzyme in the correct orientation for the chemical reaction to occur. That is kind of the point of what is happening. And enzymes are just biological catalysts. Or to help with the catalysis, they need to they lower activation energy, and so their part of that is going to be to orientate things just by the position of the amino acids which are present, and the chemistry of those amino acids are going to help to orientate the substrate and things into the correct conformation and position, steric complements in order for the reaction to be catalyzed and to move forwards. This hasn't got a red star like other things. This is really just largely just flavor. Iron sulfur clusters are going to come up a lot in oxidative phosphorylation. I just thought I'd just start with introducing it a little bit now. Step 3 is the oxidation of that isocitrate that we just generated. It's going to be the oxidation of that to alpha Kg, alpha ketoglutarate. So oxidation of isocitrate to alpha ketoglutarate, catalyzed by the enzyme IDH, which stands for isocitrate dehydrogenase. And can you remember what dehydrogenase enzymes do? In most cases, the dehydrogenase enzyme catalyzes the reduction of NAD or NADP to the reduced form NADPH from the oxidized form to the reduced form. And in doing so, this that gets reduced and this gets oxidized. So we're coupling together these redox reactions, these redox couples. And then this is a common occurrence that dehydrogenases are redox enzymes that very often take electrons from a substrate and put them onto another onto the cofactor. These often nucleotide cofactors, such as NAD, FAD, things like that. There's a whole bunch of them. But again, it's the oxidation that occurs is another oxidative decarboxylation. So it is indeed the loss of CO2. So go from the 6-carbon isocitrate to the 5-carbon alpha ketoglutarate. This is the first NAD generating enzyme in the cycle. There are different versions of this. There's isocitrate 1, 2, 3, and maybe others as well. And they differ in their localization, but also what may be the cofactor, which gets the nucleotide cofactor, which gets reduced, whether or not it is NAD or NADP. Remember that NADP is often used for other things unrelated, not as related to energy, such as cholesterol synthesis or antioxidant activity, whereas NADH is generally in energy production. And so depending on which one the cell needs, you can have the different isoform of isocitrate dehydrogenase generating one of the reduced nucleotide cofactors. But the reaction is generally the same to go from isocitrate to alpha ketoglutarate by oxidative decarboxylation, thus generating CO2. There isn't much on this slide. This is actually the only slide for this enzyme. Again, we could go into detail about the mechanism, but I'm not going to for time purposes. But if I erase the ink, just really as a summary, some key things I'd like to take away is, of course, that with this reaction, what is happening is that the electrons from the oxygen are being given to this bond, and in doing so, the electrons from this bond are given to this bond, and you get the CO2 coming off, but you get the formation of a double bond between these two carbons. Also, in doing so, you generate these two negatively charged oxygens. And these negatively charged oxygens, they don't want to be there. They are, unstable, uncomfortable. It is an unfavorable energy state. And so, again, what the things do enzymes do? They lower activation energy. Well, if you didn't have the enzyme, then these oxygens would not want to go into these negatively charged states quite so freely. But what the enzyme does is that it provides a manganese cofactor, which acts as a counterion, and thus, whereas it might require a lot of energy in order to generate these two negatively charged oxygens, because you have a cation, which is acting as a counterion, that thus lowers the activation energy to get it to a point where it may potentially occur. And then at the end, you get the rearrangement of the enol intermediate to the carbonyl, which is the alpha ketoglutarate. There's the enol intermediate to the carbonyl to generate alpha ketoglutarate. So step 3, isocitrate. Isocitrate to alpha ketoglutarate, generating CO2, generating NADH, because it is a oxidative decarboxylation. The enzyme is IDH. Those are the key, major things. You have this manganese cofactor as well, which helps lower the activation energy by acting as a counterion. This is some kind of key points, but I'm not going to test too hard on this enzyme. Similarly with enzyme number 4, again, I'm not going to test super hard on this enzyme, but I will describe the steps here as well. There will be some material, but not too much. So in this enzyme, there's another oxidative decarboxylation, taking the alpha ketoglutarate from the previous step and the oxidative decarboxylation to succinyl CoA and generating CO2 again because it is a oxidative decarboxylation, but again, generating our second NADH. And this carbon here is oxidized again and comes off as CO2. But what happens along the way is that this is coupled to a reduction again of NAD to NADH, whereas this alpha ketoglutarate succinyl CoA is an oxidation to get this redox couple again. During the reaction, the thiol of cofactor A, so another cofactor A comes in as a cofactor, comes in and attacks this carbonyl carbon again, acting as a nucleophile, attacking the delta positive carbonyl carbon to generate the succinyl CoA, which is a thioester. Oh, there it is, thioester. I was going to write it out. And this is important because this is again another energy conserving step. Energy conserving. Because you get conserving energy in that you're oxidizing the carbon and generating NADH. So that's one way you're oxidizing, you're conserving energy. But the second way is in forming the thioester succinyl, the CoA. That is energy conserving because of the high reactivity of thioesters. They don't have the same kind of resonance stability, so they're highly reactive, more reactive than esters. And so because of that, they make good leaving groups, and so the energy barrier to breaking that bond is lower, and it can yield more energy in later steps. So it actually turns out to be an energy conservation. To summarize again, again, I could talk for a whole lecture about this enzyme and go into the mechanism, but we're not going to. The key factors really is that we are the oxidative decarboxylation of alpha ketoglutarate, which is a 5-carbon metabolite, to the 3-carbon metabolite succinyl coenzyme A, which is an oxidation. And of course, the decarboxylation, you get the loss of the CO2, which is why you go from 5-carbon to 4-carbon. But the oxidation is coupled to a reduction of NAD to NADH, and energy is conserved in the reaction of the thiol of cofactor A, the carbonyl carbon of what was alpha ketoglutarate, to generate the succinyl CoA thioester. That's the key part. One additional thing which I will mention about this enzyme, I don't know if I'm not sure if I named it properly, clearly. It is alpha ketoglutarate dehydrogenase complex, and it is that dehydrogenase complex is again very similar to the pyruvate dehydrogenase complex, PDH, that I mentioned previously. And you can see that it's actually, the mechanism is quite similar in that you're causing this oxidative decarboxylation each time generating, so you loss of CO2, each time generating NADH by the reduction of NAD, each time generating the CoA adduct. Our one here is this middle one that we've just talked about. This is the one we talked about last time. And this is one that we'll talk about a little bit when we talk about amino acid catabolism, right at the end of the last thing that we talk about. But, to generate the CoA product, But in each time, it's very, very similar. This group that's highlighted here in this peach color is very similar between the different the products are all the same. It's just really this end bit that's different. But that doesn't really kind of matter too much. It's actually the main bulk of the reaction is the same, and that's because they are all the enzymes that do these reactions are all related. It's the same. If you remember from PDH, it's got the E1, 2, and 3 subunits, and funnily enough, those subunits are all very similar, likely because of a divergent evolution. What likely happened is that there was one precursor enzyme, first of all, and there was a duplication event to create several of them, and so there were multiple copies of this enzyme within our DNA earlier on, like a precursor enzyme. And then what happened is that separately, these enzymes evolved to have a different ability. So maybe there was some sort of dehydrogenase complex enzyme that existed, first of all, that catalyzed one type of reaction, but it duplicated into 3 times. And each time it created these 3 potentially identical duplicates of these 3 identical enzymes. But then what happened is that independently, they evolved different functions and became PDH. Became BCKDH, things like that. So that's likely how these came about. Of the 3 subunits, E1, E2, and E3. E3 is pretty much the same between the different complexes. E2 is a little similar, a little different, and E1 is a little bit more different. Still, they are homologous. And that means that they have some similarity in what they do and their structure, but perhaps the sequence is quite different. And the sequence may lead to a similar structure, but there is some difference there to a lesser or greater degree. But that difference in sequence is what evolved to give them their specific function of reacting with these different substrates and catalyzing these different reactions. So that's I wanted to link these together because they are very similar enzymes with similar functions. Now we'll move on to enzyme number 5. We're racing through these. We're taking our succinyl CoA, and we're going to move into succinate. So enzyme number 5 is going to break that thioester bond of the succinyl CoA to form succinate. It uses the enzyme succinyl CoA synthase here, going from succinyl CoA to succinate, breaking the thioester bond. There it is. Now, remember that I said that that again, there's not too much on this. In fact, there's one more slide on this, which is just flavor. That's just kind of an interesting thing. I just want to really summarize what is happening here. Remember I said that this thioester bond is highly reactive because it can't form thioesters can't form the same resonance stability that esters and amides do. Because of that, the barrier to breaking that bond is lower, and so that makes it easier, lowers the activation energy, makes it easier for the chemical reaction to occur, and the breakage of this bond and the hydrolysis which occurs releases enough energy to drive forward the phosphorylation of GDP into GTP. Of course, the cofactor A is released to react with something else. So in summary, and let me clear the screen. In summary, this thioester bond is highly reactive, and it means the barrier to break it is low, so there's low activation energy, and the energy released from the hydrolysis drives forward the phosphorylation of GDP into GTP, So it acts as a kinase. Kinases are things that cause phosphorylation. In this case, the unusual phosphorylation of the nucleotide diphosphate to the triphosphate. And in doing so, the hydrolysis generates the carboxylic acid succinate. So from succinyl CoA, succinate, catalyzed by succinyl CoA synthase, and you get the generation of GTP along that reaction because this is so reactive. Small thing to mention, I think is just kind of interesting in the next slide. Sometimes there's a small throwaway question on this, just because I think it's kind of interesting. If we look at the reaction, the succinyl CoA binds first of all, and you get the reaction. Let's move away from that. The main thing is, sorry, the main thing is that you generate this phosphorylated histidine. This is the bit that I'm really, really interested in because phosphorylated histidines are unusual and rare. And phosphorylated histidines are unusual and rare. That's the main thing I want to talk about. And really what you do is you find them in some unusual and primitive prokaryotic life. And that suggests, and of course, these TCA cycle, glycolysis, these things are very well conserved and very, very ancient. And so you find ancient life, these artifactual ancient life has got a lot of these, not a lot of, but some of these histidines that can get phosphorylated. But they're very rare in higher life in animals, and eukaryotic life in particular. And so that suggests that this enzyme and this mechanism here is very ancient and hasn't changed very much because it uses a very ancient type of phosphorylated histidine. And I didn't really want to talk about the mechanism, actually. I don't want to get lost in the weeds of the mechanism of the hydrolysis of this thioester driving the phosphorylation of the carboxylic acid across here, and then the hydrolysis of this phosphoester bond to generate the succinate, which causes the phosphorylation of the histidine, which can then phosphorylate GDP into GTP and restore the non-phosphorylated histidine. I don't want to get into the weeds of that. I just really wanted to mention that this phosphorylated histidine is kind of cool because it's rare in eukaryotic life, but it's something that happened in ancient life and suggests that this mechanism, this enzyme, is ancient and hasn't changed much, and that we call that conserved, evolutionary conserved. We're going to move now on to step 6, where we take that succinate and it is converted into fumarate in step 6. Step 6, oxidation of succinate to fumarate. Again, we got our friend dehydrogenase. So we know with dehydrogenase, we're going to be catalyzed some kind of reaction where a nucleotide is reduced to its reduced form, so oxidized to reduced, it's a reduction. But in doing so, you get an oxidation. The oxidation of succinate, which results in going from the alkane to the alkene. Yeah. I don't have too much, again, on the mechanism of this, which I'd like to go into. The enzyme that catalyzes the dehydrogenation of succinate to fumarate is succinate dehydrogenase. FADH2 is generated this time. That's really one of the main things. But the succinate, of course, is getting oxidized. The carbon is getting oxidized in this reaction. So you get the loss of electrons. Remember, oxidation is loss, so the loss of electrons to generate hydride ion. And this hydride ion is supplying electrons to FAD generating FADH2. And of course, what is the carbon this molecule loses electrons, so it's an oxidation is loss. And this hydride, this ion here, has got this extra electron, which is used to reduce. Reduction is gain, FAD to FADH2. That's the main thing. But something really that I'd like to mention is that this enzyme, I'm going to clear screen, this enzyme, SDH is complex 2 of the electron transport chain. So when we move on to the electron transport chain, we see that it's complex 1, complex 2, complex 3, and complex 4. And these complexes are catalyzing only 1, 3, and 4, but overall catalyzing the pumping of protons across the inner mitochondrial membrane. And we're going to go into that in a lot more detail later on. But what is happening is that complex 1 receives electrons from NADH. That's where the NADH that we're generating goes to complex 1, supplies electrons, and that supply of electrons and the movement of electrons, just like the movement of electrons through an electrical circuit can cause a motor to spin or something like that, protons get pumped across. And what happens with complex 2 is that receives electrons from FADH2, which is going to actually doesn't pump protons, it just receives them but passes them to complex 3, which then does some pumping of protons. I almost started drawing an arrow there, but complex 2 only receives but passes to 3, which does the pumping. Nonetheless, it receives electrons from FADH2. And the same thing is that why have an enzyme that generates FADH2 and then shuttle that FADH2 to complex 2? Why don't you just have complex 2 is a part of that multi-protein complex is the succinate dehydrogenase. So complex 2 of the electron transport chain contains the enzyme. Part of that complex, that multi-protein complex, is succinate dehydrogenase. I'm going to skip past this slide for a moment. I'll come back to it in a moment. So succinate dehydrogenase, where succinate is oxidized to fumarate and FAD is reduced to FADH2, the reaction happens here at subunit A. But FADH2 passes the electrons through subunit to subunit B, C, and then D. And then from D, that's where it goes to complex 3 via ubiquinol. But that's something that we'll get onto later for the pumping of the protons. But the main thing is that complex 3 of the electron transport chain is a multi-protein complex, and one of those proteins is the catalytic center of succinate dehydrogenase that catalyzes the oxidation of succinate to fumarate, and it just passes the electrons along through the complex. The slide that I skipped past is just the one that I was saying. Complex 1 receives NADH. Complex 2 receives FADH2 and via the oxidation of succinate. Protons are pumped with complex 1. Both complex 1 and 2 pass those electrons to ubiquinone, generating ubiquinol that passes it to complex 3 that pumps protons, and then that gets passed by cytochrome C to complex 4, which also pumps protons, and then it finally gets accepted by oxygen to generate water. You're not going to be tested on that yet, but just to give some context, right here, so the TCA cycle joins here, right? The TCA cycle joins up here. This is the TCA cycle, and it joins up here. Now step 1, 2, 3, 4. We can go through it, and the succinate dehydrogenase, which is step 6. So 3, 4, 5, 6, 7, 8. So it all kind of joins up together and is associated with this with the next step, the electron transport chain. And briefly, that this is an integral protein. Integral means embedded in a membrane. Okay, that was a lot to take in, but that's it. We're running out of time, but I am going to just do the next two steps, which aren't too bad. Step 7, we've generated that fumarate. So we had the let's go. So we had the succinate, generated fumarate. The fumarate, via the enzyme fumarase, we generate malate. And just get hydration across this double bond to generate the alcohol. That's it. That's it for step 7. Step 7, hydration of fumarate to malate. From the alkene to the alcohol via the enzyme fumarase. That's it. That's all we need to mention. Just think about it. Got a lot of negative charges here in the transition state. What ions do you think might be hanging around in the active site? Kind of start thinking about that. And the 8th and final step is the 3rd and final oxidation of malate to oxaloacetate, which is that metabolite from step 1 of the TCA cycle that combines with acetyl-CoA in step 1 to generate citrate. So we're back full circle, oxidation of malate to oxaloacetate. You get across at this carbon here, you get the oxidation of this carbon. This carbon is oxidized from the alcohol to the carbonyl. Sorry, guys. And this is coupled to the reduction of NAD to NADH, from the oxidized NAD to the reduced NADH via the enzyme malate dehydrogenase. Malate dehydrogenase. L is reflecting that's going to be highly stereospecific. It's only going to work on the L isomer, stereoisomer. 3rd, NAD generating enzyme. And because oxaloacetate, though it's not super favorable to occur, the biochemical delta G, the low concentration of the oxaloacetate by LCP pushes the reaction forwards to the right. And yep, like I say, regenerates oxaloacetate to go around with another cycle. That's just about it. I'm just going to wrap up with, there's a couple of slides just to wrap it up, but that's mostly it. So I mentioned that the citric acid cycle, oh, where's my pen gone? Citric acid cycle, oh, you know what? Let's go back to here. So we went through all of the steps. Oxaloacetate and acetyl-CoA, citrate synthase, condensation to citrate, rearrangement to isocitrate with a conatase, then the oxidative decarboxylation to alpha-ketoglutarate, oxidative decarboxylation to succinyl CoA, each time generating an NADH and losing the CO2 to go from 6 to 5 to 4 carbons. It then gets the generation of a GTP, because this thioester that's generated is hydrolyzed to succinate, and that reaction, the energy released from that reaction is conserved in the generation of an ATP. And succinate dehydrogenase, which is the same as complex 3, generates an FAD that feeds into complex 2, I apologize. Feeds into complex 2, generating the FAD feeds into complex 2, and that generates fumarate, which is hydrated to malate, which is then a fresh dehydrogenase reaction to restore our oxaloacetate and dehydrogenation generates our NADH, and we can go around the cycle again. So to wrap up, citric acid cycle only generates one ATP, right? That GTP that can be interconverted in ATP easily, but you do pull off a lot of electrons by oxidizing those substrates, those metabolites from acetyl-CoA. And in doing so, you generate NAD and FADH2. And that generates a lot of ATP because each NAD generates about 2.5 ATP, and each FADH2 generates about 1.5 ATP via donating electrons to the complexes pumping protons, and then those protons coming back down and generating ATP via the enzyme ATP synthase in oxidative phosphorylation. So by, which is what we're going to go on to later, but by putting those electrons into NADH, you can use them for a different reaction, which is going to generate a ton of ATP. That's the point of it. Overall, there are multiple steps in glycolysis. Where are we up to? Glycolysis, the linker step, and then Krebs cycle, TCA cycle, multiple points at which you generate a bunch of these NADH and FADH2, but also a small amount of ATP, especially here. The net 2 ATP formed, which can be repeated for fermentation. But overall, generating all of this NADH doesn't generate a ton of ATP. Only really, you invest 2 ATP, you gain 2 ATP, you gain another 2 ATP, so you don't gain an awful lot of ATP, but you do generate a lot of NADH that eventually generates a ton of ATP. So it's better to, rather than generating ATP directly, you don't get a lot of energy ATP from that. Instead, by generating NADH and shuttling that into OXPHOS, you then indirectly can generate a lot of ATP through OXPHOS, ETC and OXPHOS. I've already mentioned this hub of intermediatory metabolism, and that's just saying that in the TCA cycle, some things can come out as cataplerotic reaction, generating various organic molecules that we need, fatty acids, amino acids, purines, which can be used for DNA, porphyrins and heme, which is used in, hemoglobin and things, pyrimidines, which are, again, for nucleic acids. So a bunch of times you pull out of the TCA cycle because it's just a collection of handy reactive metabolite carbon chains that can be turned into other things that may be important for biological processes. So we pull out by various cataplerotic reactions, but we can replenish by various anaplerotic reactions. Key, of course, being pyruvate generating oxaloacetate, also perhaps glutamate into alpha-ketoglutarate, which isn't here, pyruvate into malate by malic enzyme. A few of these, which I will move on to later when we talk about amino acid metabolism. But I would like you guys to be familiar, and you probably will be tested on the idea that you can put in and out of the TCA cycle, because there's useful things that you want to pull out and turn into other biological molecules. And then you're going to have to replenish those. Or when it comes to turning pyruvate into oxaloacetate, turning that into PEP, and then going from PEP, that glycolysis substrate, into glucose, because you can't go directly from pyruvate to glucose. You need to go via oxaloacetate to PEP to glucose via gluconeogenesis, which is exam 2 material, not exam 1 material. This is all exam 1 material. I mentioned a few of these anaplerotic and cataplerotic reactions already, so I'm going backwards a bit here. Like for instance, aspartic acid. Aspartic acid can be converted into oxaloacetate, or glutamate can be converted into alpha-ketoglutarate. Alanine can be converted into pyruvate. Glutamate can be converted again into these various transaminases by taking this amine group, turning it into a ketone. You can go again, the amine group, turning it into a ketone. These transaminases are key, some of the key things. There's lots of things that can do this. These transaminases are key things which can interconvert amino acids into various TCA cycle intermediates in anaplerotic reactions, as may be. And the TCA cycle is what we call amphibolic, which means it can both go in and out. It can be part of catabolic or anabolic processes. It can build things or it can break things down as feeding out of and into the TCA cycle. Like I mentioned, we can't convert acetate, so from acetyl-CoA into glucose. We can't do that because 2 carbons come in and 2 carbons come out. The only way to, what we can do instead is convert pyruvate, which is just before it, into glucose via oxaloacetate with the enzyme PC that we'll move on to later on. But I mean, other things can. The glyoxylate cycle is where you can turn acetate into carbohydrate, but we don't have it. Simpler life does. So we can't convert acetate. We can't do the glyoxylate cycle to convert acetate into glucose. We can convert pyruvate into glucose, but not acetate into glucose. We don't have those enzymes. But overall, TCA cycle is an amphibolic pathway. And I think this is this pyruvate carboxylase, which I mentioned. We're not going to talk about we're talking pyruvate carboxylase with gluconeogenesis, but you know, just introducing that you're generating oxaloacetate from pyruvate via this PC enzyme, which may be useful for gluconeogenesis or various different other processes. And oh, sorry, one last thing is to mention is that it's very important that the TCA cycle is very highly regulated because, of course, you want it to be able to switch on and switch on depending on the energetic needs of the organism. And so various different enzymes such as PDH, citrate synthase, IDH, alpha-ketoglutarate dehydrogenase, they have regulation points where, for instance, they have allosteric regulation, where a buildup of things later in the cycle, buildup of energy, ATP, NADH, which is signaling we already have a lot of energy, they will be negative allosteric modulator of these enzymes to switch them off and slow down their activity. Or later metabolites are negative allosteric modulators to say, hey, slow down. Whereas positive allosteric modulators are going to be things like ADP and AMP, which is signaling kind of used up ATP, saying, hey, I haven't got much ATP. I've got a lot of ADP, saying that my ATP has been used up, turned into ADP. A lot of ADP will bind to these enzymes, act as a positive allosteric modulator to say, hey, let's ramp up production. Classic example being PDH kinase, but which is positively allosterically well, the kinase, which phosphorylates it to its inactive form, is positively allosterically modulated by things which are signaling a high amount of energy. But we ran out of time. But it is important that you know that these can be regulated by positive and negative allosteric modulation, in that things signaling high energy are going to bind and switch the enzyme off. Things that are signaling low energy are going to bind and switch the energy on, switch the enzyme on effectively. But that would be that. If you have any questions, please come and see me. And we'll be in class soon. Have a great day.